The next speaker is Larry LOO from NTU and IMCB A*, Breaking the Wall of Insulin Therapy for Diabetes Treatment.
In the 16th century, an Egyptian physician noticed a mysterious disease which causes the patient to experience frequent night urination. And this urine often attracted ants and often tastes sweet. Today, this mysterious disease is now what we know as diabetes. Diabetes is a condition associated with an abnormally high amount of glucose in the blood. As of today, there are no cure for diabetes, and the only way to manage diabetes is through the use of diabetes drugs and insulin injections. The prevalence of diabetes is rising. There are around 422 million diabetics worldwide, and this number is expected to rise up to 600 million by the year 2040. Imagine the global impact. Diabetes can also lead to other hosts of diseases such as cardiovascular disease, stroke, and kidney damage. And the question we ask is, why does diabetes develop? This is an image of the human islet that we published in this journal. I would like to bring your attention to the little precious green cells here as shown. These are the pancreatic beta cells, which are responsible for secreting insulin required to bring the blood glucose level down. If these cells die, the patient will eventually develop diabetes. So the problem here is really a lack or absence of insulin-secreting beta cells. To break down the wall of insulin therapy, we need a powerful tool. And this tool is in the form of the human-induced pluripotent stem cells, HIPSCs. In short, they are the eternally youthful cells which can give rise to any other cells in the human body. First, we obtain the mesenchymal cells from human volunteers and reprogram these cells back into HIPSCs using defined proteins. We then treated the HIPSCs with a wide variety of growth factors over a month-long experiment, and we was able to successfully generate beta-like cells in a culture dish. Most importantly, we also showed that these beta-like cells were able to express over 300-fold increase of insulin transcript. This is a key indicator that they are indeed the insulin-producing beta cells. Our next plan of experiment is to transplant these beta-like cells into a diabetes mice model. Our goal is to first show that we are able to cure diabetes in mice. Our advantage is that these mesenchymal cells are immune privileged. So beta-like cells made from this source would be able to avoid immune attacks by the body's own immune cells such as the CD4 positive T cells. Using this method, we are also able to generate limitless supply of beta cells in vitro. And using this entire platform as a sledgehammer, we hope to break down the wall of insulin therapy in diabetes treatment. Thank you. Okay, actually, we do stain them with, for example, the little green cells you see just now, we stain them with the insulin antibody. So if they appear green under the microscope, we can confirm that these are actually the beta-like cells.
Okay, thank you. I think you're going to have to take your questions to him later.